Krebs cycle, citric acid cycle, or tricarboxylic acid cycle. This process have 8 steps. During this process, the formation of 3 NADH molecule, and we got 1 ATP molecule from this step, and formation of FADH2 molecule. During this process, we capture NADH molecule and FADH2 molecule because this process important for the electron transport chain. Mostly student knows about that formation of NADH2, NADH and FADH2 molecule, but they don't know about what's the enzyme that's present in the Krebs cycle. I want to explain you here that in Krebs cycle, acetyl-CoA oxidized and form of citrate. Citrate isomerize. In this process, there is isomerization. 6 carbon molecule converted into the isocitrate. In the third step, isocitrate carboxylase means there is a carboxylation. Carbon dioxide molecule goes outside of the cycle, known as decarboxylation. In the presence of isocitrate dehydrogenase enzyme, NAD converted into the NADH molecule here, and isocitrate to alpha ketoglutarate. Again, the process known as decarboxylation. Two processes. One is decarboxylation, other is dehydrogenation. In the dehydrogenation process, electron transport from one place to another, and in the carboxylation, when carbon dioxide goes outside of the cycle. Alpha ketoglutarate, in the presence of alpha ketoglutarate dehydrogenase enzyme, conversion of NAD to NADH molecule and carbon dioxide goes outside. Acetyl-CoA, alpha ketoglutarate to acetyl-CoA conversion. This process also known as fifth step. Fifth step, okay? And again, acetyl-CoA convert into succinate. Succinate are sixth step. In the presence of succinyl-CoA dehydrogenase enzyme. In this process, GTP molecule converted into GTP molecule, and during this cycle, ADP converted into the ATP molecule. We got here 1 ATP molecule. Succinate produce FAD2 when succinate in the presence of succinate dehydrogenase enzyme, conversion of fumarate, and in this process, FAD convert into the FADH2 molecule. There is dehydrogenation or hydration. There is hydration means I want to say that we got H2 molecules. Fumarate. Fumarate is a 7th step. Due to this step, there is formation of malate, and malate, because fumarate oxidized, and malate converted into the oxaloacetate in the last step. It is the last step, and in the last step again formation of NADH molecule in the presence of malate dehydrogenase enzyme. Guys, so guys, there is a short information in front of you, and I want to explain about overview that how the enzyme involved in the Krebs cycle and how the 8th step works for the Krebs cycle process. If there is no formation of acetyl-CoA, no Krebs cycle. From where the acetyl-CoA produced? It produced from the pyruvate. From where the pyruvate produced? From lactic acid. If there is deficiency of lactic acid, no Krebs cycle. Thank you so much, guys.